All right.
You sure you don't want it to be completely dark? All right, so thank you very much for having me speak at your bar shop seminar today. I'm going to talk to you about some of the work that's been ongoing in the lab for about the last 2 years, where we're just now starting to see a few results and bring them in. So I welcome a discussion with you about my work and how it might influence aging, and I especially welcome any opportunities for further collaboration with the aging research community. So just a slight correction. I'm not talking too much about TBI today. I'm going to talk more about stroke, but a lot of the mechanisms involved in stroke and traumatic brain injury are quite similar. In fact, I have several ongoing projects in the lab. I'm going to focus today on the role of LRP1 in recovery after stroke and TBI, and I'll talk a little bit about the role of LRP1 in neural stem cells after stroke. I also have, so generally I'm interested in recovery after brain damage. I also have a project involving spinal cord injury, which I won't be talking about today. So to get started, I think it's always helpful to preach about why astrocytes are cool. So this is kind of a cruddy cartoon of an astrocyte here, but I think what's notable is you can see in my cartoon, it has a projection up at the top of the screen. In purple, what it does is it wraps fine processes around neuronal synapses. All right, and so this is an example of an electron micrograph in which the astrocyte here in blue is wrapping around the neuronal synapse, both the presynaptic and postsynaptic density. All right, and this is notable because in this spot, what astrocytes do is they remove extracellular potassium, water, neurotransmitters, and glutamate. They similarly release several trophic factors that are very important in maintaining neuronal and synaptic health, and this includes things like energy substrates, neurotransmitter precursors, growth factors, antioxidants, and also lipoproteins, which I'll talk a little bit more about today. Now, on the bottom of the screen here, we have a big foot process that is wrapping around the vasculature. And here, so this is an immunohistochemistry, and in red we have astrocytes that are labeled with GFAP, and in green we have a protein called aquaporin, which is localized to vascular processes that wrap around. And we can see this here when we zoom in this greenish yellow. This is actually astrocyte aquaporin that is wrapping around a small vessel in the brain. So here, what astrocytes do is they play a very significant role in helping maintain the blood-brain barrier integrity. They are the primary influxers of glucose into the brain. They help modulate cerebral blood flow. So, of course, I'm giving a seminar to the aging research community. I should talk a little bit about how astrocytes influence aging, both normal and perhaps pathological. And so this paper just came out this year, which is a very interesting paper in which they analyzed the expression, and these are from several postmortem brains of human patients. This is like hundreds to thousands of samples. And they just analyzed the expression of several factors compared to younger samples. And what they found is that glial cells, that includes microglia, astrocytes, and oligodendrocytes, tend to have a significantly greater change in gene expression with aging compared to neurons. Additionally, glial gene expression is better at predicting age than neuronal gene expression. So altogether, this just really shows that astrocytes undergo significant changes with aging, and this also means that astrocytes are probably a great target in aging research if you want to target the brain phenotype with aging. So some of the things that astrocytes specifically do with aging, and this is something that the people that were in the neurobiology of aging course already saw a little bit of. So astrocytes don't actually increase in number with age, but they do change in morphology. They tend to become hypertrophic, so they get large. They actually gain a slightly more reactive phenotype. Other things that they do is they respond less to neurons. And so in this is an example of a figure in where neurons were stimulated, and then the calcium signaling was measured specifically in astrocytes. And what this group found is that in older samples from older patients, or older, I think it's older mice actually, what they found is that there is less synaptically induced calcium signaling in astrocytes as a result of age. And another thing that happens to astrocytes with aging is that astrocytes have reduced capacity to engage in this glymphatic clearance of waste in the brain. And so the glymphatic system, if you haven't heard of it, it's a fairly new system that's been described over the past couple of years. And what this is through aquaporin 4, which is localized in astrocytes, there is a flux of liquid through the brain that is very important in clearing waste and other sorts of products from the brain. And we can see here in young mice, if we label the flux of fluid through the brain in young mice, there is a significantly large amount of fluid that is moved through the brain, right? Compared to old mice where that is not really happening. So generally, we can say that some of the age-related changes in astrocyte function include having increased reactivity and possibly inflammation. They have decreased responsiveness to neurons, and they have decreased contribution to waste clearance. Exactly, taking out the trash. I like that.
Circadian swelling and all of that.
For sure. Yes, sir.
When you refer to lymphatic waste, is that generally things that are of smaller molecular size, or can it also be consistent of larger proteins?
So the question is, when I'm referring to lymphatic waste, is it smaller or larger molecules, correct, such as proteins? Or generally, I believe it's a little bit of both. But I think there's probably I don't know for sure, and I'm not sure whether it's been tested what the upper limit on size is. Certainly true for smaller molecules, and even for things like amyloid beta and stuff, up to a certain molecular weight. So overall, with this background of astrocytes, this leads me to the lab's central hypothesis, and that is that if you can target astrocyte function, you can benefit multiple neuroprotective processes, both in normal and pathological aging. With this in mind, I want to start to tell you about one of our projects involving stroke research. Before we go too far along, I think it's worth recognizing some of the members of my laboratory that have engaged in a lot of the work I'm about to show you. That includes this undergraduate, Gustavo Salinas, who did a lot of the behavioral assays I'm showing you. My graduate student, Sadia Ahmad, who just recently successfully defended her research proposal. So I guess she's officially a PhD candidate now. And then, of course, my two great laboratory technicians, Pam and Shane, and then all the funding. Okay, so when we talk about stroke, most people know that one of the primary risk factors for stroke is aging. And one of the worst consequences of stroke is a long-term disability. In fact, stroke is the primary cause of long-term disability in adults. And when we're thinking about what happens during a stroke, I think it's helpful to understand some of the processes that occur after a stroke happens. So as we know, brain cell metabolism is relying heavily on the delivery of oxygen and glucose from blood. And when that blood gets blocked, as in the case of an ischemic stroke, what happens is obviously the disruption of nutrients causes a disrupted metabolism. And that causes something called a necrotic core. And this is what we think of as sort of ground zero for a stroke lesion. And if we zoom in on that area, which I'll show you in a minute, you can see what I mean. Surrounding that necrotic core is something we call the penumbra. So if we zoom in and look at that, the core here is in this darker brown color. And this really nothing survives because there is just not enough oxygen and glucose to support life in that area. But surrounding that area is something called the penumbra, where it is at risk to die. And in fact, the core can expand into the penumbra. But this is also an area that is a place that you can target and stop some of the spread of this injury that occurs after a stroke. So some of the cool things, well, cool scientifically, not cool if you're the one who has a stroke that happens, is one thing is we have cytotoxic edema. And this is an example from Jim's lab, Jim Leckleder's lab, in which they were filming in real time astrocytes that are labeled with GFP, and they induced a focal stroke in a vessel here, and then they just watched the astrocytes, and you can appreciate that over the course of this stroke, the astrocytes which are there pop like little balloons, and then the GFP is going away. And so this is this is the necrotic lesion, this is the core where everything basically explodes and creates sort of a toxic soup that helps spread the injury into the penumbra. Yes, ma'am.
What would be happening to the microglia and oligodendrocytes in this kind
They're very likely doing the exact same thing. So we see neurons exhibit, so their axons exhibit like beating, and that's kind of how they exhibit cytotoxic edema. I've not personally looked at microglia, but I would not be surprised if the same thing happens there. Yes?
With that last one, so with the penumbra, but we don't see reactive astrocytes in the penumbra. Does that come later, much later in the? Because I mean, when you look at a stroke, you get this huge amount of reactivity.
So we've seen increased in GFAP, which is one way to help measure reactive astrocytes, as early as 24 hours. But that's kind of it's not nearly as robust as later time points. And so if you look at it at 2 weeks over, there's significant glial scarring that is very obviously appreciable.
So how long is this video?
This video right here, this is within minutes after the stroke. So this is happening very quickly.
So,
Yeah.
if astrocytes are responsible for moving material from the synapse back to the bloodstream, like potassium, is that what they're doing? They're taking excess potassium and moving it back to the bloodstream?
Well, so they're not necessarily moving it to the bloodstream. Certainly that's one place they can go, but so they can take it up, and then they actually, through gap junctions, can buffer that potassium into nearby astrocytes, and then once the ion homeostasis is reestablished, they just return to normal function.
I'm just thinking about when you induce a stroke,
Mm-hmm.
now you no longer have any glucose, so you no longer have any energy source, you've got no place to get the electrons, you don't have oxygen. What's going to happen? You start accumulating a lot of potassium in the nerve cells that are in the area of the stroke.
So that's a fantastic question. So his question was, what happens when you don't have any energy? You can no longer buffer the potassium or take it up effectively. And what happens is you have basically excitotoxicity and spreading depolarizations, not just in the region of the stroke, but because axons can extend very far out, they can extend across the entire brain. Seizures is one side effect, and all sorts of nasty things. Okay, so another thing that happens after a stroke, which I had the movie going here, is microglia respond to the injury. And so this is just an example. This is not a stroke, but this is very similar to what happens with microglia. And this is actually a laser-induced injury. And the microglia, what they do is they immediately react to the damage, and they actually extend processes toward the injury. And the idea here is they're going to be some of the first defense in walling off that damage, and they're also going to be removing a lot of the debris from the necrotic core. Okay, so these are the things that occur in the very acute stages after a stroke. And all of these things actually contribute to spreading of the damage. So much like everybody's scratched their finger or their arm or something, and you maybe start with a tiny scratch, but after a few days it seems like the scratch has gotten bigger. That's called a secondary injury. And what happens is the primary injury causes processes to occur in nearby cells that result in their cell death. Okay.
So does potassium go up in the CSF after the stroke?
I am not sure if potassium goes up in the CS. I would not be surprised, but it probably also depends on the severity of the stroke. Okay. So while this is the first opportunity for treatment of a stroke, the question is, what happens after the stroke is ostensibly healed? So the size of the lesion is no longer spreading, okay? Maybe the stroke is stabilized, but there are several processes that continue after a stroke. And some of these things include scar formation and inflammation, and I'm calling this the chronic secondary injury phase, okay, in which you don't have significant growth of the original lesion, but you have ongoing processes that might contribute to brain pathology. For instance, and this is an example of a glial scar that happened 2 weeks after a stroke, in which we have significant astrocyte hypertrophy around the area of damage, and they're actually helping wall off some of that damage. You also have inflammation, so this is penetration of peripheral macrophages and other cells into the brain, but also activation of resident brain inflammatory cells, such as microglia. And then, altogether, one of the things that can also happen as a result of all these ongoing processes is neurodegeneration. Okay, and this is an example of a brain that had been injured well in the past, and it has increased neurodegeneration as shown by fluoride jade staining. So stroke does contribute to poor outcomes in long-term processes. So patients that had no previous neuropathology have double the risk of cognitive decline if they have a stroke. 30% of stroke patients that have recurrent strokes, they actually experience dementia with aging. So I'm very interested in understanding what are some of the factors that might contribute to a poor long-term outcome after stroke or other brain damage, and importantly, why those factors might contribute to a poor long-term outcome. And if we start to think about some of the factors that might contribute to a poor long-term outcome, right here, I'm thinking of if we think sort of a teeter-totter or a continuum, and at one end is dementia and at the other end is perfectly normal aging, there are some things that are going to push that teeter-totter closer to dementia, right? And some of those things might be lifestyle choices, such as, being obese or smoking. Aging, which you can't really help, will also perhaps push you closer to that edge. And then other things, like if you're just born unlucky and perhaps you have different mutations, might also contribute to dementia with aging. One of those notable genetic factors that can influence the likelihood of getting dementia with aging is Apolipoprotein E4. So patients who express ApoE4, they have a greater risk of coronary heart disease, ischemic stroke, and cerebral microbleed. So altogether, their vascular health is worse. And patients with cerebral palsy, so these are usually patients that had a perinatal stroke, okay? If they carry ApoE4, they tend to have epilepsy, or they have to use a gastrostomy tube, which means that it was likely that perinatal stroke actually was more damaging to them than somebody who didn't have ApoE4. If you carry ApoE4, you tend to have a worse functional outcome after stroke, traumatic brain injury, or brain hemorrhage. And if you're a mouse carrying ApoE4, you tend to have greater midlife cognitive impairment if you stress that mouse as a child. And finally, ApoE expression in general increases as you age. So ApoE, and particularly ApoE4, seems to be detrimental in many brain conditions, and it increases with aging. So this suggests that expression of ApoE4 can significantly increase the possibility of cognitive decline with age, and this would be particularly true in patients that have a history of brain damage. So what is ApoE4? We know it equals bad. So what is it? Well, ApoE4 is a lipoprotein, and its primary responsibility is to carry lipids. In the brain, it carries extracellular cholesterol around the central nervous system. And it's important to note that in the brain, cholesterol homeostasis is separated from that in the periphery. So the cholesterol your liver makes does not get into your brain. Instead, your brain has to rely on cholesterol synthesized and secreted by astrocytes. And as you age, neurons rely more and more on astrocyte-secreted lipoprotein, which is one of the reasons it increases with age, because astrocytes become less and less capable of synthesizing their own cholesterol. You can also imagine that cholesterol actually makes up about 20% of the brain's weight, which is significantly greater compared to your cholesterol in the rest of your body. So your brain is significantly reliant on the production of cholesterol. So apolipoprotein E actually has 3 allelic variants. We have ApoE2, which is associated with a decreased risk for dementia with aging, although you have increased cardiovascular disease. ApoE3 is considered the wild-type allele, and so this is about 75 to 80% of the population have this. And then ApoE4 is associated with both an increased risk of cardiovascular disease and an increased risk of dementia. Dementia. 2 dementias. All right. Okay, so the main thing about ApoE4 is it's really just caused by a mutation in a single amino acid right here, from cysteine to arginine, number 112. And what that does is it can do a lot of things that will affect ApoE function. It changes the way that protein is able to carry lipids, and it also decreases the stability of the protein, and it very likely alters its ability to bind to specific receptors. And so what happens, as I was mentioning, astrocytes will secrete a package of ApoE, and so this is a discoidal package of lipids and ApoE, and as it floats around the CNS, it actually acquires a cholesterol ester core, and so it becomes a big fat ApoE, which then moves around and helps traffic cholesterol to different cells, significantly here in neurons. Okay. Now, in order to get used by the cells, it has to be removed from the CSF via something called receptor-mediated endocytosis. So if we zoom in, and I'm zooming in on the astrocyte because I prefer those, although this would happen in any cell in the body. Receptor-mediated endocytosis is actually Nobel Prize-winning work by Brown and Goldstein at UT Southwestern. And what they describe is lipoproteins, and in fact several other sorts of ligands can do this too. Lipoproteins will bind to a receptor, which then causes the receptor to be endocytosed into the endocytic lysosomal system. And once there, the cargo is sorted for degradation and the lipids can be trafficked to other membranes in the cell. And then the receptor then can be returned back up to the plasma membrane where it can continue its job as before.
So it has to endocytose both the ApoE and the cholesterol as a unit.
Correct. Cholesterol is generally poorly soluble on its own. Okay, so how is ApoE removed from the cerebrospinal fluid? It is removed most predominantly by low-density lipoprotein receptor family members. And so the one we're most familiar with, and the one that won the Nobel Prize here, is LDL receptor, right here. And this is like the tiny receptor in the family, okay? And we have several other receptors here, and notably is this one here called LDL receptor-like protein 1, or LRP1. So obviously, if we're just comparing the structure, LRP1 has significantly larger extracellular domain, and this is reflected by the fact that it can bind promiscuously to a whole series of different ligands, which I'll get into a little bit more as we go along. So Miranda, when you age, LRP1 does things. So here, actually, there's two important domains in LRP1. There is the transmembrane domain and there's this extracellular domain. And so as you age, you actually have decreased levels of this plasma membrane domain of LRP1. This is also correlated with Alzheimer's disease patients. So if you have Alzheimer's disease, you're more likely to have decreased expression of LRP1. Now, the extracellular domain of LRP1, perhaps conversely or perhaps it makes sense, tends to have greater expression in the CSF, or actually it's disconnected from the plasma membrane, it's proteolytically shed, and it's found in the CSF of older patients and patients with Alzheimer's disease. So altogether, it does look like that with aging, LRP1 expression is altered, and it could be a target for things that are occurring during aging. Correct. It is shed by, I think, atom proteins, proteinases. All right. So in addition to the fact that it changes with aging, okay, and it influxes ApoE into the cells, it serves as a clearance receptor for membrane proteins on the plasma membrane. It clears amyloid beta from the cerebrospinal fluid, and it also clears several ligands floating around the CSF. So this is a partial list of some of the things that LRP1 is known to interact with and remove either from the extracellular space or from the plasma membrane. So it's a gigantic list. But the question I have then is, it's clear that LRP1 has some pretty important functions within the cell. So could it be possible that ApoE4, by virtue of its altered binding and altered behavior in the body, could that interfere with the ability of LRP1 to perform its other normal important functions, such as binding and clearance of other proteins? And there is some evidence for this in the literature. So if you have ApoE and amyloid beta, they will actually compete with each other for clearance from the CSF by LRP1. So LRP1, which I didn't mention, is the primary clearance receptor for amyloid beta. And if you add ApoE to that mixture, you actually have a reduction in the clearance of amyloid beta. If you add ApoE4, and if you, let's say, infuse it into the brains or whatever, it will increase that proteolytic shedding, what I mentioned of that extracellular domain, compared to other ApoE isoforms. So my hypothesis altogether then is that ApoE4 causes LRP1 to have altered association with other proteins, and this would disrupt the cellular response to injury after stroke. So this is a general explanation for how ApoE4 might alter LRP1 activity and therefore cause possibly a spectrum of different issues in the brain. So obviously we can't study every single thing that LRP1 potentially does, but we can start to focus it on a few potential targets. And to do that, I grouped some of the processes that I think are important and occur after brain damage, right? Some of these include the blood-brain barrier disruption that occurs after a brain injury, the astrocytes response, neurons and whether or not they survive after the injury, ongoing inflammation, the functional recovery of the brain after damage, and then finally things that might influence ongoing neurodegeneration. All right. So to start, I picked the astrocytes response and inflammation because inflammation likely plays a very significant role in long-term ongoing brain damage. So one notable thing from the literature is that LRP is able to regulate the surface expression of the TNF receptor. And this is actually in peripheral cells. So this is in endothelial cells and macrophages. And here they just have some cell lines. And on the right, they have wild-type cells, and on the left, those that have knocked down LRP1, as you can see here. And they have increased surface expression of the TNF receptor. So my question is it possible that a similar regulatory event might occur in astrocytes? And if so, how might this influence a long-term outcome after stroke? Additionally, can ApoE4 mimic a phenotype of LRP1 deficient cells? So here, if we examine this more closely, we're thinking of one of the important roles of LRP1, which is the internalization of ApoE4, and another important role of LRP1, which is to remove this TNF receptor from the plasma membrane. The real question is, in the presence of ApoE4, does it somehow negatively influence LRP1 activity such that it impairs its ability to remove the TNF receptor from the plasma membrane? And in that instance, if there's more TNF receptor on the plasma membrane, that means that the cells could be more sensitive to cytokine signaling, and that in turn would cause greater sensitivity to brain damage. So this is important because if you have prolonged cytokine stimulation, particularly of the TNF receptor, that can lead to apoptosis. All right. So the hypothesis here then is that ApoE4 can increase the brain sensitivity to TNF-alpha by decreasing the ability of LRP1 to regulate the TNF receptor. And if you're not sure, I suggest that this is probably a bad thing after brain damage. So the first thing we do is we start to look at these in cells. And so the question we ask is, does the loss of LRP1 then increase the sensitivity to TNF-alpha? What we did is this is actually an immortalized astrocyte line. There is no ApoE in these cells, so these are ApoE null. We received these from David Holtzman's lab, and we created a stable cell line that has persistent knockdown of LRP1. And we take those cells and we can do some mechanistic inquiry. And so here what we do is we just want to get an idea of what LRP null cells do before we add in ApoE to see if it mimics the phenotype. So we stimulated these cells with TNF-alpha for a period of time, and then we just measured the stimulation and whether or not, and the signaling that occurs after that. And so what we find is that here are the knockdown cells on the right, and on the left are the wild-type cells. We actually stimulated with a pretty low dose of TNF-alpha. We use 2 nanograms per mil. Typically when people do these experiments, they use 50 nanograms per mil. And what we found is we had pretty low activation of TNF-alpha signaling cascades as measured by phospho-NF-kappa-B in wild-type cells here on the left. And compare to those that don't have LRP1, we have significantly greater activation of this signaling pathway. And of course, we have it quantified down below. But another thing that I think is helpful in looking at these is if we're looking, so we see that there is greater activation of the TNF-alpha response. We also see that that activation is more prolonged. And so if we compare this as a maximal response, right here. So 100% is set as the maximal response. What we see is that the wild-type cells decrease back to baseline levels, their signaling over time pretty quickly. So this is sort of a negative feedback response from TNF-alpha. By comparison, those that are lacking LRP1, first of all, they already have a higher baseline of this signaling activity, but they also take more time to return to baseline. So if we're thinking of this as a signal, it's a more prolonged signal as well. It's both stronger and more prolonged. Excuse me. So what is some of the functional consequences of this prolonged activation of NF-κB and signaling pathways? So one of the first things we started to look at is cell viability. And so in this instance, what we did is we took our same astrocytes that we had before, and we measured the cellular viability after prolonged treatment with TNF-alpha. And what we find is in cell culture, we actually did this with 2 different clonal cell lines for each expression level. And what we found is that up to 10 nanograms per mil of TNF-alpha doesn't really kill wild-type cells, but as little as 2 to 5 nanograms per mil managed to kill cells that are lacking LRP1. Or maybe I shouldn't say kill, there's a loss in viability. So whether that's due to decreased proliferation or actual cell death, we haven't tested yet. Another thing that we wanted to look at is, this is starting to get at what part of this protein is influencing the phenotype we're seeing, is does inhibition of LRP1 ligand binding alter TNF-alpha sensitivity? And so one of the ways we can do this is actually use something called receptor-associated protein. And this is a chaperone that is actually co-synthesized with LRP1, because LRP1 is so sticky that if you don't add the chaperone, it never gets out of the ER because it ends up binding everything and then it just turns into a mess and it gets degraded. But the nice thing is this is a great tool because RAP then goes ahead and gloms onto all of those receptor binding sites on LRP1 and then can inhibit that extracellular domain of LRP1. And so what we did is we treated with this receptor-associated protein for a period of time, and then we stimulated with TNF-alpha, as I just showed you, and measured the viability as before. And when we do that, what we find is that RAP pretreatment does not alter the cellular viability in response to TNF-alpha. Now, we still need to do a very important control, which we're still figuring out how to do, which is making sure that our RAP is actually working to inhibit LRP1. But it does potentially suggest that this extracellular domain of LRP1 is not actually important for this phenotype that we're seeing as a result of LRP1 knockdown. And so the question is, if it's not this extracellular ligand binding domain, maybe it has something to do with this 85 kilodalton plasma membrane binding domain. And we are looking into that more. So I've showed you some of the effects in cells that don't have LRP1. What happens when we add ApoE to the mix? So these experiments are pretty preliminary because we're just now getting good at actually purifying lipidated, nice ApoE3 and ApoE4. But I can give you some of our preliminary results and see what we have here is we actually use mammalian synthesized and secreted ApoE. And the reason we use this rather than, say, a recombinant protein, I'm looking at you, Dr. Craig, is, is that, yeah, right. So if you're using a recombinant protein that is expressed from bacteria, it is not properly lipidated. And the lipidation of ApoE plays a significant role in its function. And so really the only way you're going to get properly lipidated and expressed ApoE is getting it from a mammalian system. And so what we do is we have HEK cells that have been stably transfected with either ApoE3 or ApoE4, and then we can then purify that lipid from that extracellular fluid, which would be media, conditioned media. Okay. And so what we do, and these are some preliminary experiments, but we can take our ApoE null astrocytes, and then we can pre-treat with the ApoE. We can use ApoE4, but as a control, we'll use ApoE3, or we can even use the null conditioned media, so we kind of control for all of those conditions. And then we stimulate with TNF-alpha, and we measure the phospho-NF-κB, the activation of that pathway. And preliminary results seem to suggest that if we add ApoE4 into our experiment, this increases the sensitivity to TNF-alpha. And as I mentioned, these are preliminary results. This experiment was not actually done with the as well purified ApoE as we are doing now, but at least preliminary data seems to suggest that it increases TNF-alpha signaling. Similarly, we can now look at the dependence on LRP1 of this effect, right? So we do the same experiment, but now we do them in astrocytes that are lacking LRP1, all right? And so the hypothesis there would be, if there's no LRP1, adding ApoE4 should not increase the sensitivity to TNF-alpha. And again, preliminary evidence seems to indicate that without LRP1, ApoE4 does not seem to exert an increase in TNF-alpha signaling. So altogether, the preliminary conclusions from these experiments seem to say that LRP1 can decrease astrocyte sensitivity to TNF-alpha if it's expressed. That LRP1 modulation of TNF-alpha signaling does not seem to involve that ligand binding domain. And that ApoE4 seems to increase the TNF-alpha sensitivity in a LRP1 dependent manner. So I'm going to move forward to some mouse experiments, but before we go on, is there any questions that people want to ask about this particular part? Hi, Miranda.
What is the expression of TNFR1 in your LRP null astrocytes?
We're still trying to properly compare that to wild-type cells, so that we're working on that. It seems like overall the levels might be fairly similar, but what we need to look at is the surface expression. And so those experiments are ongoing.
So how is it that LRP1, so LRP1 decreases TNFR1 expression?
Yes.
Does it form a dimer?
Yeah, likely what it's doing is forming a dimer and then escorting it into the endocytic system. Yes, sir.
Well studied, the LRP1 expression in the neuron is much, much higher
Correct.
than the astrocytes. I wonder how's the distinguishing that's not involved in the activation of TNF?
Right. So so the question is, LRP1 is actually significantly greater expressed in neurons compared to astrocytes. In these experiments, we're just looking at astrocytes, but we're going to get into later on some things that we see, and your point becomes much more valid then in that because LRP1 is expressed in all sorts of cells, the response we're measuring in astrocytes may not tell the whole story that we see in animals. And the question is, we don't know.
Similar to this question is, of course, the glials express LDL receptor. It's much more than the neuron in the reverse. So is it also the LDL receptor have such similar function or not?
So LDL receptor is probably a good control to look at. So the difference between the two is that LDL receptor doesn't have promiscuous ligand binding. LDL receptor is pretty much good at binding lipoproteins, and that's about it. So one would predict, although it's worth testing, that if you knock down LDL receptor, does that affect signaling in this capacity either? Perhaps if it did, that might suggest that the problem is not a receptor-specific problem, but maybe a membrane composition problem, in that the lipid composition is somewhat altered by not being able to import cholesterol, would be a guess. Yes, sir?
Which is what I was just going to ask. If you've got a cell at 75 kilodalton clip left in the membrane, do we have a clue what happens to that? Does it get recycled? Is there anything yet?
So it is, so that...
Does it turn out to be a signal? Metabolic signal?
Right, and we don't know for sure. We do know that domain seems to play some roles in signaling, so it can actually help propagate signals by dimerizing and helping recruiting things to a signaling molecule. Beyond, and it probably can also escort proteins to the endocytic system. Whether or not by proteolytically shedding the extracellular domain, does that decrease the stability of the C-terminal domain? I'm not sure. I would say it would make sense.
The next question I'm going to ask then is, what if it's even more powerful than a neuron, then what does that have to do with tau? Or if anything, it has to do with tau.
We don't know what it might have to do with tau, but I think it probably has a lot to do with amyloid, but we're not ruling out that it has something to do with tau. And in fact, because it interacts so heavily with ApoE, which seems to now possibly influence tau aggregation, there's probably some sort of crosstalk.